I think you can all see my slides, so let's get started. Um, so, um, the story of drug discovery today is a tale of so first a word from our general counsel, and now let's dive in. The story of drug discovery today is a tale of the glass half full and the glass half empty. This is a bit of a depiction of the glass half full, where medicines that have been discovered over the past 50 years have taken diseases that were a death sentence or a sentence to a life of pain and have transformed them into chronically managed treatments or sometimes even cures. Vaccines, of course, are very much in everyone's minds today, but there's so many other diseases that have been meaningfully addressed by our growing ability to interrogate biology and develop medicines that leverage that understanding. So that's the glass half full. The glass half empty is really well articulated by this graph known as Eroom's law. All of you are familiar with Moore's law, which is an exponential increase in productivity of technology. Eroom's law, the inverse of Moore, is an exponential decrease in productivity in pharmaceutical R&D, where the number of approved drugs per billion US dollars has been going down steadily and exponentially year on year. You'll notice that the y-axis is on a logarithmic scale, and the large factor in that decrease in success and increase in cost is the fact that the vast majority of the drugs that we put into clinical development, 95% or more, depending on how you count, never end up making it to an approved drug. And as a consequence, the cost per approved drug has to carry on its back all of the many, many failures that never made it out. And so it ends up being an amortized cost of $2.5 billion and rising. So why are there so many failures? There's so many steps in the journey between when one begins with a concept of let's work on this aspect of this disease through to that holy grail of an FDA approval. And that journey involves multiple forks in the road, each of which has many, many paths that we can take that don't end up leading anywhere and cause long digressions and possibly even the failure of the entire program. And oftentimes we have no idea which one to pick. And it is all those wrong decisions that build up on each other to give us that 95% failure rate. So what if we could leverage machine learning at different stages in the journey to make better decisions and take the right path a bit more often than we're taking today? So a main place of failure is that we need to understand what disease even is so that we can appropriately focus on the right elements of addressing whatever it is that was the disease-causing factor. Today's taxonomy of disease is oftentimes 50 plus years old and based on the coarse-grained clinical manifestations that are oftentimes subjectively filtered through the lens of a patient and a clinician, and have very little to do with the underlying biology of the disease. If we're better able to interrogate the underlying biology in a human that gives rise to these disease manifestations, we might be able to identify patient populations that really do have a coherent biological mechanism that drives the disease, and hopefully then better identify nodes that are intervenable within that process. So in order to address that need, we're able to leverage a technological revolution that's happened in biomedicine in the last 10 to 15 years, which is the ability to collect incredibly rich and large amounts of data about human biology at unprecedented fidelity and scale. Those data come up both in humans, so we can collect from human patients things like MRIs of entire body systems, histopathology data at very high resolution, blood biomarkers of different kinds, thousands of those, and those give us a view on human biology. At the same time, we can juxtapose that with data that we can generate in a lab where we can take human-derived cellular systems and measure those biological phenomena similarly at incredible fidelity and scale, and get an understanding of what disease processes look like in a lab system, which also allows us the ability to interrogate interventions and see what they do to that cellular state. The challenge, of course, is that while these data can today be produced at this very large scale, the human mind is incapable of discerning the relevant patterns in those data sets, and we're not able to make maximum value of those. So machine learning, of course, is a way to address that, and we can then undertake this understanding of machine learning of this biological state, align it with clinical outcomes on the one hand, genetics on the other, and basically bridge between the genetic factors that are oftentimes causal of disease to those clinical outcomes via this understanding of distinct biological states. So in the platform that we've been developing for 3.5 years at In Citro, we really are trying to execute on this vision, collect very large amounts of data, both in vitro and from human clinical samples, use genetics as a way of creating an alignment between the what we can do in a dish and what we can see in a human, and use that to build a manifold of disease state. So we have an extensive in-house capability on our wet lab side that involves multiple elements of microscopy, cell generation, transcriptomics, and various other systems. And we have partnerships that enable us access, sometimes unique access, to human clinical samples that are alignable with what we see in vitro. And then what we're doing is, first of all, building a data-enabled drug discovery company from the ground up, including the development of substantial amounts of machine learning and workflow infrastructure that enables us to do the kind of data analysis work on these types of data in the way that allows machine learning, modern machine learning, to shine. From the analysis emerge three types of insights that are helpful, that are beneficial to patients: novel targets that are drivers of disease or modifiers of disease, biomarkers that allow us to identify the right patient populations, and of course, molecules that go after those targets in those patient populations. So today I'm only going to be able to really focus on one of the work streams that we've had because of lack of time. So I'll tell you about our work in metabolism fibrosis. This was driven by a collaboration with Gilead and NASH. And then I'll very briefly mention some of our work in neuroscience. So NASH is an acronym for non-alcoholic steatohepatitis. It is a disease that is driven by what is called NAFLD, or non-alcoholic fatty liver disease. NAFLD is when you have lipid droplets that make their way into liver cells, hepatocytes. And when that happens, the liver cells become inflamed, they die, the liver becomes inflamed, and ultimately becomes fibrotic and scarred, and oftentimes gives rise to hepatocellular carcinoma. The prevalence of this disease is very much on the rise because of the prevalence of obesity and metabolic syndrome worldwide. So in this work that we did with Gilead, we had access to a really unique data set of clinical trial data from patients across multiple trials, where we had for those patients histopathology data from liver biopsies at the beginning and the end of the trial, RNA sequencing from those same biopsy samples, as well as various blood biomarkers, and we had whole genome sequencing data for a subset of those patients. So what we did is we applied machine learning to this high content biomedical data in order to identify causal processes that underlie disease. So we started out with what the clinician looks at when they look at these data, and specifically the clinician, a pathologist, looks at the image biopsy, the biopsy of the image, the image of the biopsy, and annotates that with 4 ordinal scores corresponding to different biological processes that they have been trained to look for: fibrosis, inflammation, and so on. Those scores are quite coarse-grained and give only a very limited view of the extent to which the disease process in liver has progressed. So what we did is we took an unsupervised, or rather a self-supervised approach that took that same biopsy image and created an embedding that really captures the underlying semantics, if you will, of liver disease. And then that embedding was then used in multiple different ways to enable us a much finer-grained understanding of disease processes. And I'll talk about that in a moment. So first of all, validating the model itself, we took that self-supervised embedding and layered on top of it a single linear classifier, a single perceptron, and use that to predict the pathologist score. So this was the only supervised component of the model. And you can see that there's a very strong correlation between the ordinal pathologist scores on the x-axis and the machine learned scores on the y-axis. This is just one exemplar of that. We did that for the other scores as well. Now you can say that there's variation within each bucket assigned by the pathologist relative to what the machine learning assigns, this falls in a range. So we actually tested whether that variation within that range is meaningful meaningful. And we see, and there's different ways to validate that. What's shown here is the fact that people who are higher in the range are more likely at the end of the trial to have progressed to the next level. People who are lower in the range are more likely to regress, which suggests that what we're finding is biologically meaningful variation within that range, within the bucket that the pathologist classifies. So that's a nice validation, but it's not really, our goal really isn't to just replace the pathologist. That's really not the point. The point is to identify more interesting molecular aspects that are disease associated. So what we did here is to basically see whether we can predict the RNA sequencing measurements from those same biopsy samples. This is useful both in itself because RNA sequencing measurements are typically not collected from livers in the standard of care. So having that molecular understanding is useful there. But more importantly, it also allows us to validate the fact that our model is producing meaningful predictions of deep down biology as measured by these transcriptional profiles. And what you can see here is that the machine learned scores provide a much higher accuracy prediction for those for pretty much every gene in the genome relative to the pathologist scores. And on the left is the scatter plot. All the dots are above the diagonal, and on the right is an example gene, the one that's in orange, showing that you get a much higher correlation of the prediction from the pathology to that gene expression. Now, an actual use case of that to gain insight is to use this understanding of gene expression in order to identify genes whose expression seems likely to drive disease progression. So what we did here is we looked at genes whose expression at baseline associates with this very fine-grained disease progression that we're able to measure. And what we see is that if you look at the pathologist scores, there are only 3 genes whose expression profile at baseline predicts progression from the beginning through the end of the trial, whereas on the machine learning score, because it is much more granular, 37 genes came up. 37 is actually enough to work with, and you can now start to identify pathways and processes that are drivers of disease progression. You can't do that if you only have 3 genes. And so we were able, for example, to identify particular pathways that seem to be drivers of progression, and also validate those relative to the literature at a very high percentage. Maybe the most interesting thing, though, is going back to what are true causal factors, which is the genetics of the disease. And so here we asked the question of, can you look at this notion of disease state and disease progression and find genetic drivers for those processes? That is variation in the genome that is associated either with the state or the progression of the disease. And what you see here is probably the best annotated and best validated genetic driver for NASH. It's a variation in a gene called PNPLA3. If you look at the pathologist scores, there is no association with the PNPLA3 variant. Now, you might say that is purely a matter of resolution like we had before, but that turns out not to be the case. Even the more quantitative machine learned scores, if they are trained towards the pathologist scores, do not show association with this variant. However, the self-supervised approach, because it does not anchor on a preconception by the pathologist on what matters and what doesn't, is able to demonstrate on the right-hand panel a beautiful dose response of the number of disease alleles that the patient has. Like, is there 1 copy of the disease gene or 2 copies of the disease gene? You can see that there is this beautiful dose response that is visible when you're training a model without having anchored on what a clinician knows to look for. Now, the nice thing is that we can then go back and teach the clinician about what to look for. And so what you see here is the ability of our model, which uses an attention layer to identify which tiles drove the prediction of the disease allele. And so you can demonstrate to the clinician what a the non-disease allele looks like in the histopathology level versus what the disease allele looks like at the histopathology level, which are the 2 panels on the top. And then you can also use GAN techniques to actually create a visualization where the disease allele is the only thing that varies across the sample and really highlight the differences that are purely driven by the disease allele. So this the focus of what I've discussed so far has really been on the analysis of human clinical data, which is a big part of our work. I mentioned that we also have extensive wet lab capabilities that allow us to interrogate disease processes in vitro. I'll show you just one very small example of that with a microscope that is actually a proprietary microscope that we built in-house. This is a really cool microscope that you can see in the top left side here. There's a set of LED lights that rotate and shine light into the cells at different angles. And then there's a camera that captures not only the intensity, but also the phase of the light. Now, why that's important is because as light shines into the image, what it shines through, depending what it shines through, you have different angles of refraction. So if it hits fats, for example, lipids, it refracts in one angle versus, say, if it hits chromatin or something else. And so by taking what is effectively a gray blur and using machine learning to reconstruct, you can get a much better understanding of actual internal cell morphology, including the composition of the cell. And so machine learning allows a high contrast, high resolution reconstruction of cell morphology. We can then layer another machine learning algorithm on top of that is trained by taking the cells, staining them with a fluorescent marker, and that is associated with a particular morphological component of the cell. And you can now basically artificially hallucinate stains for those cellular compartments. Now, the reason why that's really important is because you can do that then on an image from cells that doesn't require you to fix and stain the cells, which is a process that is both error prone as well as requires you to stop the experiment in the middle and kill the cells. And so what you see on the top right is basically artificially stained cells, hallucinated stains that are on top of live cell morphology, which allows you some really interesting phenotypes that are not longitudinal phenotypes that you can't measure in any other way. So this is just an example of what those cells look like. On the top is the output, is the hallucinated stains. On the bottom is real stained cells when you stop and stain them experimentally, and basically they're largely indistinguishable. So with that, we were able to now generate a cellular model for hepatocytes. And what we did is we subjected them to an environmental stressor that induces them to take up lipids in much the same way that happens in NASH, and trained a self-supervised model, in this case on those images of hepatocytes, without telling it what the experimental conditions were. And then basically, again, simple linear model on top of the self-supervised embedding. You can see that the concentration of the NASH-causing agent comes out loud and clear without a need for a supervised training for that. So that gave us, I'm not going to talk about it, gave us a cellular disease model that allows us to interrogate reversions of that NASH phenotype in vitro. And I'm not going to get into the details of that. So maybe very briefly, just to show you another illustration of the work that we're doing is the work that we're doing in neuroscience. And the focus here for a lot of our work has been on a disease called tuberous sclerosis complex, but then we're also applying it to a much more complex disease, specifically ALS. So tuberous sclerosis complex is a rare but not ultra rare disease. Its prevalence is about 50,000 cases in the US and over 1 million globally, and we believe it to be underdiagnosed. The disease is driven by a mutation in one of two disease-causing genes, TSC1 or TSC2. And when those genes are mutated and basically are leading to a loss of function, it causes the activation or overactivation of a core nutrient sensing pathway called mTOR. Now, when that pathway is overactivated, what happens is that it leads to what's called benign tumors. Benigns means they don't metastasize, but they still have significant consequences. Specifically, when those tumors or growths occur in the brain, then it leads to neurological impairment and often profound epilepsy. So there is a class of drugs, rapamycin and its analogs, rapalogs, that are approved. They are repressors of the mTOR pathway, and they are approved for use in severe seizures, but they're only effective in about 50% of patients. So the standard way of identifying potential modifiers for mTOR activation is a single biomarker readout in cellular systems called phospho-S6. It sits downstream of the mTOR pathway. And when people look to interrogate whether something is a disease-causing or disease reversion, they use the single marker, and they basically measure fluorescence of these punctate spots of phospho-S6. We took a much broader approach that really allows us to interrogate the multiple downstream consequences of the mTOR pathway. We measured multiple different downstream readouts, cell morphology, we measured single cell transcriptomics, and we used that live cell imaging that I showed you in the previous slide in the context of hepatocytes. We use that in this context as well. I'm only going to show you, in the interest of time, one of those examples, specifically that same live microscopy microscope that I showed you a moment ago. And what you see here is truly the example of live cell imaging. The x-axis is days, and the y is a disease axis that we train towards the severity, if you will, or rather the extent to which a cell looks more like a wild type cell versus more like a disease cell in which the TSC genes have been knocked out. And what you can see here is that at the beginning of the experiment, there is a very clear demarcation between the disease and the wild type genes. As we administer rapamycin over time at different doses, you can see that the disease cells become more and more similar to the wild type cells relative to this semi-supervised classifier, and you can also see a clear dose response, and that higher doses of rapamycin give rise to a more significant reversion, which is exactly what you'd like to see. With that, we were able to then interrogate multiple genes that came out of a screen in which we screened to this type of phenotype, specifically the reversion of the disease phenotype in an in vitro system. And we were able to take one of those genes that came out as one of the stronger hits in that screen and interrogate that in the context of this particular system. And you can see here that you get a very similar extent of reversion from that genetic knockout, which is the yellow line, as you get from the rapamycin treatment, which is the red line. And again, the green line at the top is basically the double knockout that is untreated. And all of this, by the way, is done in neuronal systems that we grow in-house from stem cells, so that they are relatively faithful recapitulations of the cortical neurons in which the disease occurs. So this was a very short description of our work on TSC. As I said, we've done this across multiple different modalities, but now we're working on a much more complex disease, specifically ALS. ALS is a devastating disease that causes the death of motor neurons. It begins in a focal region, but then spreads pretty rapidly, progressing to paralysis of nearly all skeletal muscles, typically leading to death within 3 to 5 years of onset, mostly from respiratory failure. It's also called Lou Gehrig's disease, so some of you might be familiar with it under that name. There's only 2 treatments for this, and they're very only symptomatic and are not fundamentally disease modifying. The good news for us is that 10% of cases are caused by known genetic mutations, which we can engineer into our cell lines and really measure those disease phenotypes in a meaningful way. And then in the same way that we did for TSC, and then try and identify, as we did for TSC, reversions that are relevant to that subtype of the disease. So the goal here is basically to take this broad landscape of known genetic drivers of ALS, which participate in multiple pathways, often in quite complex and subtle ways. Ultimately, they all lead to motor neuron death, but probably via quite distinct mechanisms. So if we're able to disentangle those mechanisms and find appropriate reversions for each of them, that might allow us to identify meaningful, coherent patient groups for which we can revert the disease. And so we're doing a lot of work in this area, have created a lot of cell lines with different genetics, ways of creating motor neurons from our stem cells, as well as all the phenotyping work that I showed you before. So maybe just to end this talk, a couple of high-level thoughts. First of all, as we think about our work in drug discovery, our goal really to discover and develop transformative medicines for patients. That's true for a lot of companies that are in the drug discovery space. But our hope is to do it not just do it, but do it differently. Do it by de-risking and accelerating pharmaceutical R&D by using predictive models that are derived from applying machine learning to data at scale. And then, in the way that machine learning has the tendency to do, our goal is to build a data-enabled flywheel that over time, as we collect more data about the same types of cells in different conditions with different genetics, we get better and better at identifying these disease-modifying treatments and are able to more rapidly find medicines for patients. So I think, do I have one more minute or should I pause now?
Yeah, I mean, if you feel free to continue for a minute. There's some questions. I don't think we'll get to all of it, but what you're talking about is so great. I just keep going for a minute.
Okay. So maybe this is just a wrap-up slide, where I really get to wax philosophical and tell you why I'm doing this versus the many other things that a machine learning person can work on these days. I like to think back on the history of science and recognize that there's particular epochs where one area of science has really moved incredibly quickly in a short amount of time because of the availability of a new way of looking at the field. And so chemistry in the 1800s, because of the understanding of the periodic table, physics in the early 1900s, by understanding the relationships between energy and matter and between space and time. Computing, of course, in the 1950s via the development of machines that are able to do calculations that only a person, and maybe not even that, had been able to do. And then in the mid to late 90s, two fields suddenly took off and almost independently. The field of data, which rests, of course, on computing, but also has elements of statistics and neuroscience and optimization, and the field of quantitative biology, by which we measure biology at incredibly large scale in ways that we couldn't do before. I think the next field that is really going to emerge is the synthesis of those two fields, in which we are able to measure biology at unprecedented fidelity and scale, interpret what we're seeing and identify patterns in it using the tools of data science, and then use techniques like CRISPR and various others to engineer biology to do things that it wouldn't otherwise do. This is going to have incredible consequences, I think, in healthcare, but also in agriculture and in biomaterials and in energy. And I think it's just an incredible field to be part of. And so I would encourage all of you who want to make an impact in the world to consider this field that I'm calling digital biology.